All right, well, welcome to Science Saturdays. Today is January 10th, and we are talking about the immune system. So, our immune system can be divided up into two major pieces, the innate or the fast-acting immune system, and then the adaptive, which is slower to respond. And with our innate immune system, these include, oh, sorry, when we speak fast, we're talking minutes to hours response time, as opposed to the adaptive immune system, which takes hours to days and sometimes even longer into weeks. With the innate immune system, those include our first responders that go after all germs, whereas our adaptive immune system are composed of specialists. So these are immune cells and components that are highly specific to different pathogens. The innate immune system is present from birth, whereas the adaptive immune system is something that is acquired over time. The innate immune system has no memory, whereas the adaptive immune system is what we talk about when we say immunological memory. The innate immune system is nonspecific, and different cells, though, do a much better job at neutralizing different pathogens. So we have some cells that are much better at taking care of viruses, some cells that are better at bacteria, and others that are better at parasites. The adaptive immune system is highly specific to its target. And the innate immune system is composed of the physical barriers, including our skin, mucous membranes, and stomach acid, as well as, oops, sorry, that is a typo there, cellular, which are the macrophages, neutrophils, eosinophils, basophils, and a couple others as well. As far as the adaptive immune system goes, it's primarily cellular, including our lymphocytes and our, which are our T and our B cells. And then we also have the antibody component. Antibodies are produced by those B cells. All right, so what does this end up looking like? Well, over here in this light pink, we have our innate immune cells. Over here in the blue, we have our adaptive immune cells. And then we have these really awesome set of cells. These are the natural killer T cells and our gamma delta T cells. And these are both a type of lymphocyte, like the T cell. And these are T cells, effectively, that have the ability to bridge between the innate immune function and the adaptive immune function. And so these are ones that are fast acting and don't or have a different set of requirements than our traditional adaptive immune system. We're not going to get too deep into the function of these because they are specialized cells and not a very big component with relative numbers as compared to some of the other cells that are in or that make up our immune system collectively. So let's talk a little bit about what these different cells do. Our monocytes are involved in surveillance and migration. And so these are the different cells that migrate around the body. They also differentiate. They can differentiate into macrophages or dendritic cells. They are phagocytes, meaning that they consume other things that could be damaged. Welcome. It could be cellular damage, or it could be something like actual parasites or bacteria. These are also antigen-presenting cells, meaning that when they go through and phagocytose or consume things, they will present the different antigens at the surface. An antigen is just a molecule that can be used for detecting or detecting that thing. And then we have the dendritic cell. Like I said, this is something that is a differentiated monocyte. There are also some other questions as to its origins. These are tissue surveillance cells, and so these are in our tissues, like our muscles and our skin. And they also migrate. They're also antigen-presenting cells. They also secrete cytokines, which are proteins that can cause inflammation. They're also involved in immune tolerance, meaning that these are cells that play an important role with tolerating ourselves. And so they're important in autoimmunity, and these may be differentiated from monocytes. Macrophage, this is one of my favorite cells. These are big eaters. Macro is big, phage is eater, and they're involved in phagocytosis. They're also antigen-presenting cells. They can be involved in inflammation, both pro and anti-inflammatory processes. and they're really big in tissue repair and wound healing. So then we have our natural killer cells. These are cytotoxic cells, and these are really great at identifying and neutralizing infected and malignant cells. So cells that might have a virus or ones that may be turning into cancer. They also produce cytokines, and they are also involved in immunological surveillance. Then we have the neutrophils. These are the most common bloods or white blood cells in circulation. These are also phagocytic cells, meaning that they gobble up other things. They are also granular cells. So all of these different spots in here are cytotoxic materials that once they're secreted can neutralize anything that's nearby. And so that could be other cells, it could be parasites, it could be bacteria, and they're also antigen presenting cells. And then we have eosinophils. These are also granular cells. These are especially effective against parasites, and parasites including worms. They are phagocytic, meaning that they gobble up other stuff, and they're also involved in our allergic response. They're antigen presenting cells, and they produce cytokines, and they are also heavily involved in tissue repair. So once tissues get damaged, eosinophils are there to help with tissue repair. And then the basophils are also granular cells. They're involved primarily in pro-inflammatory processes, including chronic inflammation. And they are potent in allergic reactions as they are histamine containing cells. And so histamine is kind of what gives you that plugged up feeling up here in your sinuses. And then last we have our mast cells, and these are similar to basophils, but they are primarily located in our connective tissue. And so like our different tissues instead of in our blood. Then we have these NK cells and the gamma delta T cells. These are innate-like T cells, which brings us to our T cells. So we have two major classes of T cells. They're the CD8 positive, and these are our killer T cells. These are the ones that are going around in our blood system looking for cells that have been infected by viruses. And these are the ones that grab on and then they release some cytotoxic materials and neutralize those cells. And so those cells don't necessarily get better, but they are killed by the T cell. So it's kind of neat. And then there are the CD4 positive helper T cells, and these are the ones that activate other immune cells, including our B cells. And these are also involved in cytokine production and release. And something that I didn't talk about with our cytokines and our chemokines, those are little proteins that are secreted by our immune cells to bring in more immune cells. And that'll make sense in just another couple slides, or why that's important will make sense in another couple slides. And so among our helper T cells, we have our Th1, our Th2, Th17, Tregs. We're discovering more and more of these different subsets of cells. So they're pretty unique. They do different things. Some of them are better at identifying intracellular bacteria and viruses because not all bacteria are on the outside of a cell. Some of them are inside a cell, like tuberculosis. We have our Th2, which help boost our B cell response. We'll talk about that next. And then we have our Th17, and these are ones that defend against extracellular pathogens. And so that would be like our worms again, for instance, or our other big parasites. And then we have Tregs. So these are regulatory T cells, and these are involved in self-tolerance. And so too many or too few can impact autoimmunity, meaning that when our immune system starts attacking ourselves. All right, so then we have our B cells, and these are the cells that are essential for immune memory. And these are completely and entirely specific to a single antigen. And so an antigen is a molecule that can bind immunoglobulin or a T cell receptor. So immunoglobulins are also known as antibodies. Those are produced by these plasma cells here and some of the different subsets of B cells. And then our T cell receptors are the ones that are expressed by our T cells. And so all of these are specific for something special. And so yeah, we'll get into that in another couple slides too. I dare not do that on this slide. B cells have a very complex differentiation and activation pathway. We'll see this at the end of the slide as it relates to transformation, so the development of different B cell cancers. These are also cells that present antigens and secrete cytokines. Memory T cells are in circulation. They initiate the rapid response and they secrete antibodies. Then we have the follicular and the marginal T or B cells. These are in the lymph, so like our actual lymphatic system, as well as our lymph nodes. The marginal are in the spleen. And both of these play an important role in identifying invaders early on. And then we have the regulatory B cells, and these are immunosuppressive. And then we have our plasma cells. And so when we think about our long-lasting immunity, these are the cells that are at least in part responsible for that. So they're the ones that are producing antibodies over the long term because these are long-lived, they're non-proliferating, and they're generated late in infection. And that's what gives us protection from getting something over and over and over again. And the role of these are to mass produce high affinity antibodies, so ones that are highly specific to whatever pathogen they target. All right, so I'm not going to dive into this outside of saying that this is where they all fit together. So what we have up here is hematopoietic stem cell, and that stem cell leads to the development of all of our different major white blood cell classes. And so we have our monocytes and our macrophages, we have our granulocytes, our neutrophils, eosinophils, and basophils, and then we also have our lymphoid cells. And so these are our natural killer cells, our T cells, and our B cells, and all of the different types of B cells that can come along. So they're all related, even though they're highly specialized at doing different jobs. So when we look at our blood cells under a microscope, so let's say that you did a finger prick, you got that blood, you put it under the microscope, you can see a whole bunch of different things. So this isn't what they look like. If you were to just do that, you have to stain them first. And so what we have right here, these light pink ones, do you want to take a guess? What do you think those might be? What do you think would be the most common type of cell that we would find in our blood? You think it might be a blood cell? Did I put you on the spot? I'm sorry. All right, so these light pink ones are actually our blood cells. So our blood cells have this really cool concave look, and so that's where this white comes into play, is that they look kind of like if you were to have a donut that got messy and they're still attached. And so it's like this really neat concave look, which is how they stain up in here. But what's really cool is that based off of the way that these different cells stain, you can tell what the white blood cells are and how many are in circulation, which can be quite informative as to whether or not there's any diseases that are taking place. And so what those cells look like is right here we have a neutrophil, and right here we have a neutrophil. You can see that the nucleus, which is this funny shaped thing in here, this is where the DNA for the cell is, are very uniquely shaped, right? And they stain similar. These are neutrophils. All right, then right here we have a lymphocyte. And whoops, goodness. And the thing about lymphocytes is that you can't tell if you're looking at a B cell, a T cell, or an NK cell without identifying what proteins are being expressed on the surface of those cells. So all we can say is that's a lymphocyte. And then down here, we have this kind of large cell. This is a monocyte down here, and you'll see that it kind of has this horseshoe-shaped nucleus. It has kind of a large cytoplasm. This is an undifferentiated cell. It's a monocyte, and these are the ones that you often find in circulation. You're not going to find macrophages or dendritic cells in circulation. It's going to be monocytes. And then these two over here are kind of interesting. Can you tell me what color do you see that makes this one special? What color is here that isn't over here? Pink. Good job. So this one stains differently because it's an eosinophil. And so this is called an eosinophil based on the coloring of the dye that is taken up by these cells. And so these have this really pretty, whoops, really pretty pink hue. Whereas this one down here is darker, but what color would you call that? It's kind of a black. It's the darkest purple that we can get, and that's a basophil. And once again, it has everything to do with these little dots here. These represent cytotoxic granules, and the materials that are inside those kind of little bubbles are what makes the stain look pink or darkest purple, right? Oops, and I did it again, flying through that. So do you see that? We can identify different cells based on how they stain. All right, and so these are your five major cells. Like if you were to go and have your blood tested, these are the ones that they would look for. So this is a white blood cell panel with differentiation. You'll be looking at your neutrophils, which, as a reminder, primary job is phagocytosis. They're immediate actors. These are your most abundant white blood cells in circulation. And for different people, that normal means a different thing. So it could be anywhere between 40 and 75%. This is why we need to go through and have blood testing over time. That way, it's a really big range, right? And so if you've got a really big range, what you need to determine is what is your own personal normal. And then we have our lymphocytes. These are our highly targeted, highly specialized cells, also with some innate immune function. These make up about 20 to 40% of our cells, white blood cells. And so these are our second most abundant white blood cells in circulation, which makes sense. They're highly specialized. You need them for immunological memory. And these are for adults, not for children. It's going to look a little bit different because remember, these are acquired over time. And then we have our monocytes, and these ones are involved in surveillance and phagocytosis and cleanup duty. And these only make up about 2 to 8% of our white blood cells. And then we have our eosinophils. These are involved in degranulation, allergies. Remember that these ones are really good at parasites. These make up less than 5% of our cells in circulation. And then last, we have our basophils. And so these are also granular cells, granulocytes involved in parasitic infections and also inflammation. And these make up less than 0.5 to 1% of our cells in circulation. And so when you go to the doctor and you have your white blood cells checked and they count and see how many total, so actual cell count and then relative expression as to one another. So these are all relative. This is the percentages relative to all white blood cells. You can tell something might be going on. And so if you have your lymphocyte count really, really high, that might indicate that you have either an infection. And so maybe you're getting sick, you're coming out of being sick. Maybe you were exposed to something but you didn't get sick. Or alternatively, that could be an indicator for cancer, like leukemias and lymphomas. If your eosinophils are really, really high, that might mean that you're dealing with a chronic allergy. Or it could indicate that you're dealing with some parasites. And so it just depends on the conditions in which maybe you went exploring abroad and you came back with a parasite because you tested the food. Sometimes that happens. And so that's why we are asked when we go to the doctor, have you been abroad or have you left the state or the country? And then same story with the basophils. And so it could be really informative as to what's going on inside your body right now. And so the important thing about these cells isn't understanding the role in which each one of them has. Like nobody needs to know that eosinophils and basophils are responsible for parasitic infections. If you can keep that in the back of your head, that's awesome because then maybe it'll help with your blood panels. But I think the thing that needs to be taken away is that injury and infection look a little bit different with the cells that are coming to that site of injury or infection. And so in our first minutes to hours, we have this really rapid influx of these neutrophils. These neutrophils then release these granules. They also secrete cytokines and chemokines, which are proteins that help recruit other cells, like our second and our third cells, to the site of injury. And these ones also gobble up all the bad stuff. So let's say you got a big old cut on your hand, sliced it right open, and you have germs all over on your hands. Some of them are just fine. They're not going to cause any type of infection, but some of them are pathogenic, meaning that they can cause an infection. They can start to generate a lot of pain, swelling, and then maybe you'll end up with some yellow pus if the infection takes off. But in that first immediate moment after the injury, you have neutrophils in your tissue. You also have neutrophils in your blood. And what will end up happening is those neutrophils that are in your tissue are going to shoot off a whole bunch of proteins. And it's going to bring more neutrophils to the area because these are our most abundant blood type or white blood cell type in our blood. And then those neutrophils are going to bring in more neutrophils. And so there's this really big cascade. And so if you end up getting a cut, what you'll see is that over time, it's going to start feeling worse, not better, right? And so if you even a little paper cut initially isn't going to feel really bad, but over time, as it swells and you get more and more water in there, it's going to hurt. And that water that's coming in is also coming in because of all the cells that are coming in. So then after a couple hours, it's going to be nice and swollen, might be starting to get hot to touch. It might feel warm without touching it. So we have all of these different things that are taking place. And this is when our monocytes are infiltrating, our tissue macrophages are getting activated, our dendritic cells. So we have this secondary innate immune response that are taking place. Now remember, these macrophages are really good at gobbling up stuff, gobbling up bacteria that could make us sick, as well as all of the tissue damage that had happened in the case of a cut. And then last, we have these lymphocytes that come in. And so these lymphocytes are less important for injury, but very important for infection because when we end up getting all of that bacteria in there, then those lymphocytes can do a good job of making sure that they have an even quicker response if you get exposed to it again in the future.
Example, where would like a scab?
So scabs, so that's an even, so with our immune cells, they're secreting all sorts of different stuff, but that is actually outside the immune system. So they're recruiting a whole bunch of different types of cells. The big part of immune cells is gobbling up the tissue damage because you can't have tissue damage for long periods of time because otherwise that might generate an immune response that you don't want. That could cause things like autoimmunity. But those are going to be our connective tissue cells. And I can't remember off the top of my head exactly fibroblasts. There we go. And so they're going to end up secreting a whole bunch of different proteins that are involved in tissue repair. And so they're going to secrete proteins that can interlock with one another and stem any type of bleeding, for instance. And so that's where your scab comes in. But it's also they bridge that opening because even in the smallest of cuts, you still have an opening, right? So your fibroblasts are going to go through and secrete a whole bunch of proteins that help close that up because as long as it's open, it can be infected. All right. So our first responders are our neutrophils, and then we have our macrophages. They clean up the damage and the pathogens, and then our lymphocytes, and this is involved in that robust targeted immunity. And what's kind of cool is this same type of process takes place when we are vaccinated. So let's say you go and get your flu shot. That initial tenderness at the point of injection, like, oh gosh, my shoulder really hurts, right? That's an influx of those immune cells, both an influx of the immune cells coming to that site, but also the immune cells that are residing there. And that is the production of inflammatory cytokines. So if you end up with a very tender shoulder, that's to be entirely expected because you just had an injury take place there. And then we have that swelling and then more immune cell infiltration. And so when people talk about how they feel fine immediately, but then, like later on in the day, they're absolutely wiped out, that's a sign that the immune system is properly working. It's doing its job the right way. Now, lack of that response doesn't mean anything bad, but having that response isn't a bad thing. And then a reminder that these lymphocytic responses take time. So these lymphocytes take a long time to generate that response. And so until they start generating things like antibodies and having that specificity to whatever was just injected, usually it's some sort of viral particle, you are in fact susceptible to infection. And so that's why physicians recommend that you have a flu shot several weeks before the start of flu season, is because it takes time for that immunity to happen. And so a lot of people have the idea that, once my shoulder feels better, oh, look, I'm all immune. And then they get upset because they come down with the flu a couple days later. Generally speaking, that's because you're still susceptible to it. It's not because it gave you the flu. And so that's one of the big misconceptions about vaccines. So just touching on that note. And so that actually leads us into the discussion of natural immunity. So, immunity that you get by being exposed to a virus itself versus vaccine acquired immunity. So, what is the difference? So, for this, we are going to break this down. I know it doesn't look like a lot of fun, but here we have a virus. This virus attaches to a cell. The viral DNA goes in. It's going to produce a whole bunch of virus. But if this is a phagocyte, then what ends up happening, or an antigen presenting cell, then what ends up happening is that viral DNA ends up getting broken down and or viral proteins for that matter, any type of virus molecule. So it could be a protein, it could be sugar, so it could be a string of sugars. Anything that seems specific to that virus itself will be broken down into what are called antigens. And then these antigens get docked in these human proteins and then expressed on the cell surface. And no matter what, and then they interact with our lymphocytes. And so this is what happens in an antigen presenting cell. So this could be our macrophages, our monocytes, our dendritic cells, our natural killer cells, anything that can present antigen. So if it comes in contact with a virus, it can shoot part of the virus molecule up to its surface to activate our adaptive immune system. And what's really cool is that's how we gain immunity in natural ways. But vaccine-acquired immunity is generated the exact same way. And so in this case, we can have an attenuated virus, which is a virus that has reduced pathogenicity. And so these are the ones that make you feel a little bit icky for a little while, but otherwise, like, hey, we're good to go. Or it could even be the different viral particles. And these are completely and entirely non-infectious. And so there's no way you can get sick with the actual virus itself with using viral particles because there's nothing that is functional here. This is just pieces of a virus. And then it goes through the same processing. And so they have the same mechanisms, but vaccines will get you there without a full-fledged sickness. That's one of the biggest advantages of vaccine-acquired immunity is that you get that immunity without having to be exposed to the virus and then actually deal with being sick that whole time. And so, speaking of natural, there are some major risks to infection. And so, one of the big ones that's really interesting right now is alpha-gal syndrome and meat allergy. And so, this is a tick-borne disease, but it has an unknown vector. We don't know if it's something that the tick has in its saliva, which has been characterized as being kind of venom-like, or if it might be a parasite, a bacteria, or a virus that is being released when the tick bites you. But what it ends up doing is resulting in an allergy to red meat. And I kind of chuckle, and I shouldn't, because I believe that somebody passed away from anaphylactic shock. It was previously a nuisance, but now it is a bit more serious. So what this ends up looking like is that you have this tick. The tick bites, something happens. Once again, we don't know what this is. This could be the venom, it could be a virus, bacteria, or a parasite. We just don't know yet. And this is a relatively new thing, and this is the way science works, is once you identify something, then you spend a lot of time trying to figure out what's going on. But what we do know is that alpha-gal glycoprotein, so this is a protein that has sugar molecules on it, and alpha-gal glycolipid, so this is a lipid, so a fat that has sugars on it, both have these different pathways that end up resulting oops, sorry, missed that one. It ends up resulting in this cross reactivity. And so these basophils, remember, these are part of our allergic response. So these really potent because they have histamine, end up producing these antigen presenting complexes that are similar to what is found in red meat. So we have this cross reactivity that takes place. So these basophils are inappropriately displaying these different antigens that are non-specific to the tick, and therefore we have this cross reaction that takes place with red meat. Does that make sense? It's when your immune system is doing too good of a job, but doesn't have proper oversight to say, whoa, let's pump the brakes because this this isn't anything that should generate an immune response, but it does then. And so that's what we talk about when we say cross reactivity. And so, and this is something that is believed to be a mechanism in a whole bunch of different things, be it allergies or autoimmunity. But we'll end up learning a lot about cross reactivity when we dive in a little bit deeper. We, as the scientific community, not me, not us, but the science community, as we go through and do a better job of understanding what is taking place here and how that cross reactivity impacts the allergic response to red meats. And yep, like I mentioned, this is a similar mechanism for other allergies. And so let's pop into the world of cancer then. So an important thing that people probably realize but don't think about is that every single nucleated cell that contains DNA that is dividing has the potential to transform, meaning it can become cancer. So that's our skin cells, that's our blood cells, that's our, different stem cells that are in our brains. So any cell that is dividing has the possibility to transform. And each one is a little bit unique. And that's why we really haven't cured cancer yet, is because each one of those cells are unique. Otherwise, they wouldn't exist. We don't really have a lot of space for redundancy. But yeah, it makes it really interesting because you can't treat or you can't tackle the melanoma issues the same as you would like a leukemia or a lymphoma. And so, with respect to cancer, these can be broken down into three major classes. These are the lymphoid neoplasms involving T cells, B cells, and NK cells. We have the myeloid neoplasms, which involve the progenitor cells, so those cells that turn into T cells, B cells, and NK cells. But we didn't really talk about these today because they're not the ones that are doing a lot of activities with our immune system. They make the different cells. And then we have histiocytoses, which are involved like the macrophages and the dendritic cells. And to break this down, the two major things that people are familiar with our blood cell cancers are our lymphomas, which take place primarily in our lymph organs, including our lymph nodes and our spleens, and then the leukemia, which is one of our blood cancers. And so, how does it happen? So, there can be a whole bunch of different ways that blood cell cancers occur. So, this could be DNA, and so this is acquired mutations and chromosomal translocations. Our immune cells go through incredible work with their DNA in order to produce that specificity for the different pathogens. It could also be from errors during the normal immune response. This is also something that could impact things like allergies. And then inherited genetic syndromes. So some genetic syndromes have a predisposition for cancer, the blood cell cancers, and that includes things like Down syndrome. Smoking can cause a lot of different cancers. One special one that is highly linked to smokers is the acute myeloid leukemia. And then radiotherapy and chemotherapy. And so actually having received radiation or chemotherapy for an unrelated cancer sets you up for an increased risk for developing myeloid and lymphoid neoplasms. Continuing, we also have, we're going to hit on this again. Speaking of natural, there are also some major risks to infection in that some viral infections are linked to the development of white blood cell cancers. And this is human T lymphotrophic virus, HTLV1. It's a retrovirus. It can make or can lead to T cell leukemias and lymphomas. Epstein-Barr, which is a herpes virus. This one is really common. It's estimated that 94% of people have been exposed to Epstein-Barr. This can lead to Burkitt, Hodgkin, and natural killer cell lymphomas. And then we have human herpes virus 8 or Kaposi sarcoma associated herpes virus. And this one can lead to B cell lymphomas. And then additionally, we have bacterial infection. And so H. pylori is a gut bacteria that, when it is pathogenic, can lead to ulcers and also gastric malt lymphoma. I won't get into malt. It's the mucus associated or mucosal associated lymphoid tissues. And so it's a combination of your B and T cells and a whole bunch of different cells that are in your gut. All right, and then just to kind of hammer that point home that different cells have different responsibilities and different cells lead to different types of cancers. These are some of the different cancers that have been identified from different B cell maturation or B cells during the maturation and activation pathway. So when I was a postdoc, this is the space in which I was doing my research. And so with naive B cells, it can, or we have unmutated chronic lymphocytic leukemia. With our mantle cells, we have mantle cell lymphoma. And then these are all maturing. We've got Burkitt's lymphoma. So these are B cells that are in the germinal center. Burkitt's lymphoma, Hodgkin's lymphoma, or Hodgkin lymphoma, follicular lymphoma. So now we have the activation. This is the generation of a plasmablast. So this is an active B cell that's producing antibodies. This is a marginal zone lymphoma. And then we have our memory B cells and our plasma cells, which are fully activated, long-lived immune cells. And these ones are can lead to lymphoplasmacytic lymphoma and Waldenstrom macroglobulinemia. So different B cells can lead to different cancers. And this doesn't even get into some of the things like subtypes and genetics, because there are different subtypes for unmutated chronic lymphocytic leukemia. This is one of the leukemias that I was looking at. So this is the space in which I spent my postdoc, where we focused on the different CLL subtypes. And so every nucleated dividing cell has the potential to transform. And in the case of B cell activation or maturation and activation, you're dealing with a whole bunch of different subtypes of B cells, and each one of those can transform into its own distinct leukemia and lymphoma. And so when people ask questions like, why haven't we cured cancer yet? We've spent billions and billions of dollars. It's because you got to spend time understanding the actual function of these cells under the normal system, as well as what leads to the transformation event that leads to cancer. And so with that, I thank everyone for joining.